All right, it looks like it's being recorded and we're ready to go. Do you want to kick it off, Frank?
Yeah, that sounds fantastic. Good morning, everybody, and thank you for joining us this morning. Chelsea's Hope is thrilled to bring everybody together with our colleagues and partners from Ionis, talk to everybody today. What we're going to do today is hear from Ionis, and in particular, we're going to hear from Kemi Olugemo, who is a neuroscience researcher and healthcare advocate, and she's the executive director for clinical development at Ionis. Kemi completed her medical degree at the University of Maryland School of Medicine and remained in Baltimore for a neurology residency before undertaking additional fellowship in neuroimmunology and multiple sclerosis at the Maryland Center for MS. As the staff neurologist and partner in the Charles County neurology practice, Kemi introduced a number of novel infusion therapies for the practice patient, which in turn brought her in contact with clinical research organizations. That's really how she got started in this part of it. And that ultimately caused her to join the pharmaceutical industry. Her current role at Ionis is focused on advancing therapies for patients with rare and ultra-rare diseases. Of course, that puts her right in our wheelhouse with Lafora. Prior to joining Ionis, Dr. Olugemo held progressive leadership roles at Axia Therapeutics, Paraxel International, and Questcor Pharmaceuticals. She's also a long-term consultant in neurology with Forum Independent Review Board, excuse me, I kind of butchered that. So we're all looking forward to hearing from her. And I know that many of you have already sent in comments. Thank you so much for sending in your questions early. That gave everybody an opportunity to review them in advance and help to prepare to give you a more engaged response. I understand many of you will have questions and comments during the presentation. What we ask is that you include those questions and comments in the chat below. We have people who are monitoring the chat and going to be feeding those comments and questions to the right person, and hopefully you will get a response in real time. Now, as it often is the case in these types of events, we may not have time to hit every single question during the event, but the staff at Ionis and the volunteers at Chelsea's Hope will collaborate to make sure that we can do our best to get those questions answered afterwards. This webinar is being recorded, and at some point we'll have a link for people to view it after the fact. So for those folks and any of your friends or family members who weren't able to see it, they will be able to see it once we get the recording online. At this point, I think what I want to do is hand it over to our colleagues over at Ionis to move the webinar forward. Thanks a lot, everybody.
Thank you so much, Frank, and thank you to everyone at Chelsea's Hope and the Lafora patient community for hosting us today and for allowing us to give you an update on our program in Lafora disease. So we can go to the next slide. So here you'll see our agenda for today, and what myself and my team are going to do is give you an introduction to Ionis and RNA-targeted therapies, reinforce our commitment to Lafora disease and the Lafora patient community, give you a brief update on our programs, as well as our communication plan, and then answer your questions, as many questions as we can get to as possible. Next slide. The next one. So here with me today, I have Kristina Bowyer, who's our Vice President of Patient-Centric Drug Development, Jordan McSherry, Associate Director in Patient-Centric Drug Development, and Dr. Mathias Leinders, who works with me on my team in clinical development at Ionis. The next slide. Ionis is a company that was founded over 30 years ago And you may already know that our focus is on RNA-targeted therapeutics. And Ionis has a strong commitment to bring medicines to patients that are founded on the most up-to-date science. And in that regard, we've already developed three therapies for rare diseases. One of them is Spinraza, another is Tegsedi, and then the third is Waylivra. And these are for spinal muscular atrophy, amyloidosis, and a third condition. So we have, a pipeline that has a lot of rare and ultra-rare therapies that we hope to bring to the market. The next slide. So I'm sure you're aware that Lafora disease is a genetic disease. And essentially it means that it's caused by a defect in the DNA. And because of a defect, too much glycogen accumulates in the nervous system, specifically in the nerve cells, in the muscle, and the liver, and in other tissues. And the glycogen accumulates in a particular form that's not soluble, and these are called Lafora bodies, so they're large extensions of glycogen that essentially cause havoc in the body and in the nervous system. And Lafora disease is caused by known mutations in either Lafora, the EPM2A gene, or the EPM2B gene, which is myelin. These are the most common, although we do know that there are other mutations that can cause a phenotype that's very similar to Lafora disease. And in the next slide, you'll see essentially the way that we hope to target this disease, and it's through something called antisense. So we all know what DNA is. It's essentially what makes up our genetic code. And we have codes for all of the genes in the human genome in our DNA that is translated through RNA. I think of RNA as a courier, which takes a message from one place to another place. And the way the antisense therapies work is that they specifically target RNA so that they address the root cause of the disease rather than a particular protein downstream. You think about it in a way as though if you have a cut, right? So you can either address the actual, cause of the tear of the artery, or you can use a Band-Aid to stop the bleeding. And antisense therapy is something that works at the root cause rather than a protein downstream. And so in the next slide, the next one after that, you'll see that we've done as a company, we spent several years preparing to bring a potential therapy to the market for Lafora disease. And so because Lafora is ultra rare, it means that other than the disease not being very common, there's no available therapy right now, and there's a lot that's not known about how the disease course is in most of the people that have the disease. And so we've really committed to partnering with the research community to learn as much as we can about the disease, along with the patient community, and how best to target it using the type of therapy that I described. We've identified a lead drug candidate, which is ION-283, that we are trying to advance towards the clinic. We have invested in 2 natural history studies. One of them is currently ongoing. We've done a number of animal studies, and we've seen some preliminary signs that there's a potential for the therapy to work in humans based on these animal studies. And we're currently finalizing a protocol design that we can take to the clinic to test in patients for the first time. And we've communicated recently that we anticipate this first clinical trial happening in 2022, but it's unlikely to occur before the second half of 2022. The next slide. In terms of the ongoing natural history study that I mentioned previously, this study has been going well, and we expect the last patient to complete their last visit in February of next year. So what's going to happen after that? We'll have a clinical study report, and we will have publications that everyone will be able to review and get access to. The data that we put out will be anonymized, so no one will be able to identify who participated in the study, but you will get data in a summarized format. And this study is very important to help us to describe the course of the disease, to see if there's any correlation between a specific type of mutation and how fast or slow someone progresses, and to understand other factors that are associated with disease progression. And then very importantly, to help us to figure out how we can measure response to treatment so that we can design a clinical trial to give the best possible results. The next slide. So now I will hand it over to my colleague, Dr. Mathias Leinders, and he'll take you through the results of the burden of disease survey that many of you participated in, and then we'll come back to the next portion.
Yeah, hello everyone. Thank you for the nice introduction, Kemi. I personally want to, first of all, thank you personally for participating in the burden of disease survey. The data that we generated through your participation is actually immensely helpful. As you know, the burden of disease survey captures key patient experience data that helps us to develop new therapies in a better way. The types of insights that the patient community provided is helping us understand the patient journey, unmet needs, the most bothersome symptoms, and the impact of the disease on the quality of life. This type of information helps us design better clinical trials with meaningful measures of effectiveness and ultimately generate evidence that shows that a treatment is improving the lives of people affected by the disease. It also gives you an opportunity to actively be heard and influence the design of meaningful clinical trials. Regulators such as FDA support and encourage this type of work because it helps to ensure that new drugs have a clinically meaningful impact of patients' lives, especially in rare diseases where little is known. Next slide, please. And this brings us actually to the results of the burden of disease survey. In January 2021, we set out and collaborated with Chelsea's Hope to gather respondents and participants for this immensely incredible, helpful work. In total, we almost enrolled up to 40 respondents, 37 in particular, with a median age of 20 years. Most of the respondents were caregivers and indicated that 76% of people were Lafora disease patients that were Caucasian. 51% were male and 49% were female. The predominant mutation was EPM2A or Laforan, with 68%, followed by 24% of people carrying mutations in Malin or EPM2B. Next slide, please. The next slide characterizes the diagnostic journey, showing the median age of onset or the first time symptoms occurring at around 12 years of age, but the formal diagnosis being made 3 years after with about 15 years of age. This is incredibly helpful information as it shows a disconnect between the time of onset and the time of diagnosis, which is a key learning for us as it may indicate a need for improved diagnosis. On the right slide, you see invaluable data. This is an example of a respondent voicing his or her patient experience to report valuable learnings for us that go beyond what is actually reported in the literature. As you can see, the patient is saying, due to the fact the seizures, convulsions, spasms happened also in her sister, the doctor suspected it was a genetic disease and because of this asked them to do the exams. This is key learning for us. Next slide, please. The next slide focuses on common misdiagnosis in Lafora disease. 78% of respondents indicated a misdiagnosis in their journey. And on the right side, it shows us what these actually were. Why is this helpful for us? Well, for example, when trying to access more patients or improve the diagnostic process, this is an area where we all can focus on, and that goes beyond what is reported in the literature or that we can learn from natural history studies. The next slide, please. The next slide reports all symptoms that were captured. This is a key slide for us for the development of patient-centric therapies. While we see a lot of agreement with what we see in the literature or what we learn from natural history studies, that, for example, people suffering from Lafora disease have myoclonus or suffer from seizures and cognitive deficit, it also shows us symptoms such as fatigue that are very bothersome to the patients. These examples are what we learn from and what we use as a guidance for patient-centric development of therapies. The next slide, please. This slide looks at the most bothersome symptoms in a greater detail. We gave respondents an opportunity to voice what the symptoms are that they want a therapy to beneficially impact. In case for people suffering from Lafora disease, these are seizures, cognitive deficits, and trouble with motor function through, for example, myoclonus. This data is used to guide the decision and is used as a key for patient-centric outcomes in clinical development and helps us fine-tune our clinical strategy to optimally show benefit of our treatments for patients. Slide, please. And I think we're giving this over now to my colleague, Kemi, to give us a further update on the Lafora disease program.
Thank you, Mathias. So as Mathias mentioned, the results from the survey were really very helpful for us in our planning of the current trial that we're going to be initiating in patients for the first time, because we want to make sure that we are addressing the most important and the most bothersome symptoms to patients in the study design, and that we're testing the ability of our potential therapy to be able to do that. So thank you for that summary, and thank you again for everyone who participated. If you go to the next slide, you'll just get a quick overview of the upcoming activities, and I'll walk you through that. And then, we'll have time for questions, as I mentioned previously. So we're in the top left section of the slide. So we are in the process of developing and designing the clinical trial protocol, as I mentioned. And we're doing this with the help of experts in the field. So experts who have taken care of Lafora patients for quite a long time. We are also using the data from both natural history studies as well as the survey to help us design the best possible protocol. We have to seek input from regulators. And the reason is because there's no precedent. So there's no outcome measure that's existing that we know that can comprehensively capture all the aspects of Lafora disease and measure it properly. So we're having discussions to see how we can best do that in the clinical trial, along with all the other elements of designing a protocol, such as ensuring that the starting dose is safe, that the doses we test will be effective, that the study has scientific integrity and scientific rigor, and that it's acceptable for a potential registration down the line. So that's an ongoing process. Along with that, we have to identify sites, right, to run the study, and we've made some headway in doing that, and we'll definitely be calling on the sites that have already participated in the prior natural history studies that are either ongoing or completed, and we'll be using the learnings that we gleaned from those. We also, as I mentioned, have done quite a few studies in animals. These are not completed because with rare diseases and many in common diseases too, you continue to learn through your development program. So we're continuing to check, in particular, long-term toxicology studies to make sure that if the drug is given on a chronic basis, that it's still as safe as when it's given just for a short period of time. And then, as I mentioned previously, next year we'll be initiating the first clinical trial in patients. So in the next slide, you'll see some really important things that we'd like you to keep in mind. We don't know if this drug is safe or effective in humans because it's never been given to any human being before. So that's one of the key questions that we'll have in the first trial, and that we can't answer until we have some data. Clinical trials will help us determine the most appropriate dose. It's also important to know that even though we try to be as inclusive as possible, the first study will likely not include everybody, so everybody won't be able to participate. And it'll probably be a study that's small, so it's not going to be hundreds of patients enrolling in the study. Unfortunately, we cannot give access to a drug outside of the clinical trial process, for example, with compassionate use or expanded access, until we get preliminary safety and efficacy information. And that's a process that we recognize has been slow and frustrating, but we're committed to seeing it through and continuing to move along the process. And go to the next slide. In terms of keeping you abreast of what's going on, we commit to providing regular community statements as we have to give you updates. And these statements will be released through the patient advocacy groups. We will have webinars if we have something important to communicate. Perhaps Jordan or Kristina would want to say something more about that. And definitely we'll continue to use Chelsea's Hope as a resource for us and for the community. And here you have an email address on the bottom for Ionis. So please use that to contact us to ask us any questions that you may have. So again, thank you for your time today. And Jordan, if you want to say a few words before we open up the Q&A for questions.
Sure. So thank you, Kemi, for walking us through, and thank you, Mathias, for reviewing the burden of disease data. I think that everything that the patient community has done and contributed over the last several years has helped further this program. And so that's really important to know that everything that you've done, every visit to the natural history, in the natural history study, every survey, all of that is valuable and it's being used, and we appreciate it. And I'm going to kick it over to Kristina to start with the FAQs and some questions that we received, so we can answer some of your questions.
Awesome. Thank you, Jordan. One of the recurring questions that I see is just questions about like the clinical trial process and the timeline. Maybe Kemi, if you could just speak to kind of the length of a study and what and kind of how time expectations and in clinical studies are not something that happens overnight, and give people a little bit more perspective of what to expect.
Sure, happy to do that. So before you can initiate studies and patient populations, there are a number of steps that have to take place. And all those are called non-clinical or preclinical studies. And they're done in animals and in multiple species for different reasons. So the first thing you want to do is test your hypothesis to see if the drug is going to work on the pathways that you think are going to be relevant in humans. And you hope, of course, that anything that you find out in animals will be translated to humans, but it's not always a perfect fit. In fact, it rarely is. So you're testing your hypothesis. Does the drug work on the systems and the processes, enzymes, proteins, et cetera, that you think it'll work on? You look for safety in non-clinical studies. So you look to see what kind of toxicities the drug will cause, and what doses would be safe to begin, and what your maximum tolerated dose would be, so that you can calculate those in humans so that your study in humans is safe. You look at things like pharmacology. So where's the drug going? Is it going to the liver? Is it being eliminated through the kidneys? Does it, cross the blood-brain barrier and go into the central nervous system, or does it stay in the periphery? All those things are important to determine how best to ensure patient safety during a trial, as you can imagine. So along with those, you also need to get some preliminary information about long-term dosing, because you can give a drug, once or twice, and the effects can be different than when you give a drug for a very long time. All of these studies take years. They take years to perform, to review, to analyze, and to put out reports, and then to get scientific advice from the appropriate regulatory bodies. And so that's something that we've been doing along with the natural history studies and the patient burden of disease survey that I mentioned previously. And so we're coming to the tail end of that, obviously, because we're planning the trial in humans. And in terms of the duration of a study in patients, typically the first study that you do in patients is small. It's a small study. You're only looking at safety and tolerability of a drug. We'd like to do more because we know that it's so difficult to get patients. We know that the need is so urgent. And so we want to look at preliminary efficacy as well as safety and tolerability. And the regulations allow you to do that. And that's part of the nuance of what we're doing in conjunction with our advisors to make sure that we get as much data as possible from that first study. So not just safety and tolerability, but also, does the drug work? So, it's an iterative process, but as I mentioned, we've been committed throughout the way, and we feel like we've made significant gains. So hopefully that answers the question, Kristina.
Yes, thank you very much. I think another question that we've seen is just what are the inputs right now from FDA? So maybe you could kind of talk to that question a little bit as well.
I'm sorry, can you repeat that? What is the
have we gotten any input from FDA?
So we have gotten some preliminary input from the FDA, and we have ongoing discussions, and we plan to go back to get some more additional input. So that's not something that we've communicated publicly at this time because we haven't made any final decisions, but we definitely plan to continue getting input from the FDA.
And maybe you could just speak to the value of that, right? So that I kind of often explain interactions with FDA is, we want to have an agreement on kind of the work that we're doing going forward. And it's important that it's, the study that's being designed, right, is not being done in isolation. And so the value of that input is pretty critical to the program.
Absolutely. So some of the questions you might ask, in addition to verifying that the FDA agrees with your dosing, agrees with what you call a maximum tolerated dose or safe dose, is how you compare your drug. What are you comparing it to? Are you comparing it to a placebo? Are you comparing it to an existing therapy? Are you comparing it to natural history? Those are questions that you have to get answers to, because if you don't get input and you find out later on that the FDA doesn't agree, then that can really put you behind, can put the program behind, and can put potential therapies on the back burner, because you do the trial and there's no way to actually get approval and get access. So those are the types of questions that we have for the FDA, but not just the FDA, right? Because we hope that that we would get approval outside of the U.S. too. So I would put in Europe and Canada into those regulatory bodies.
Thank you, Kemi. I think that was a really important question to address. One of the other questions that I see in here are on the development of the inclusion criteria, and have those been developed yet, and how and when would we share those?
So along with the other aspects of the clinical study design, we have some criteria that we think are important to include, but they're not final. And this is also something else that we have to get agreement with the regulators on. So we know that once we have a protocol that's approved, we will be able to post that on clinicaltrials.gov, and that will be available. So the eligibility criteria, inclusion as well as exclusion, would be available on clinicaltrials.gov. So it's not ready yet, but when we have those eligibility criteria finalized, they will be available on that website.
Absolutely. And that is something that, we would want to announce in a community statement as well, and share with the community once that that posting is available. Let me go to another question really quick. There is a question about medications which might be allowed in the clinical trial. Would you be able to speak to that a little bit, Kemi?
Yeah, absolutely. So I mentioned that our plan is to be as inclusive as possible. So we really want to make it as feasible as possible for as many people to participate. So we're not going to exclude any medications that a lot of people are going to be taking, for example. Right now, because we don't have any criteria, it's really important, and even through the trial, just to follow the medical advice from your doctor, your prescribing healthcare provider. So any therapies or medications that are prescribed are done so with that individual patient in mind, and should be followed. But right now, we don't have any exclusions that I can communicate that we know for sure are going to be in the protocol.
Thank you. I know you touched on it a little bit earlier, but there's a question about compassionate use. If you could speak to when that might be an option for patients.
Yeah, so that's a very important question, and it's something that we definitely think about and is on the top of the mind of us on this team and the company as a whole. Compassionate use would not be available until we have basic information about safety in humans. And so, as you can imagine, we need that to be able to give any therapy for compassionate use. So we're going to proceed with the first study and get preliminary safety and efficacy data, and then we will be in a better position to answer that question.
Got it. There was also a question about the drug will not be ready in a few months. So maybe just reiterating the timeline might be important as well.
So we're not anticipating being able to start the clinical study in patients until the second half of 2022. And along with that, we're not going to have any drug available until we start the clinical trial. I'm not sure if that answers your question, Kristina.
Yes, it does. Actually, thank you. I think, let me go to the next one. Do we know where the clinical trials will be conducted, Kemi?
So we do know that we plan to have sites in Europe and in the US. It's possible that we would include additional countries or regions, but Europe and US is very likely. And as I mentioned previously, the sites that are participating in the current natural history study will be included in the first in-patient study.
Got it. And then there's another question about travel to another country to participate in a clinical trial. And I think, that's something that is really going to be site dependent, because sometimes individual countries and institutions have their own regulations as to where they can accept patients to participate at their site. So I think that's something that we'll just have to wait and see. Let me look for another question here. There is a question about how will the drug be administered? Maybe Kemi, you could speak to that.
Sure. So this is a drug that will be administered into the spinal fluid, so through the back in the lumbar spine. So it will be inserted. It's basically an intrathecal injection, and it's kind of like getting a spinal tap or lumbar puncture. So not going to be, a pill or an injection under the skin.
And there was also a question about having a placebo group. Do you know if we will have a placebo group at this time?
So that's a good question. So most regulators, especially FDA and European Medicines Agency, they prefer to have a placebo comparator because that is the most scientifically vigorous way to evaluate if a potential therapy works. It doesn't mean it's the only way, but that's the method that is generally accepted and looked upon more favorably. So that there's definitely potential for that to be the design, but as I mentioned, we haven't finalized the study design yet.
There's also a question about patients accessing their natural history data. Maybe you could speak to if that's going to be an option for patients, Kemi?
Sure. So we do plan to publish in an aggregate form data from the natural history studies, but this will be done in a de-identified way so that there are no patient names, there's no protected health information. You can't identify anyone that participated in the study. So that's the format in which the results would be communicated so that we can, look at how everyone did as an entire population and get, specific details as well, such as potential correlations between different mutations, between ages and age of diagnosis, what particular aspects of Lafora disease were affected. So it's going to be an aggregate data publication and not individualized.
Got it. And then there's a couple of questions in here about the stage of patients, whether the study could potentially be limited to pre-symptomatic patients, or will it include late stage late stage patients as well?
Another important question. I don't know that we've made a final decision on the entire study population. As I mentioned, we're going to try to be as inclusive as possible. What I can say is that it's unlikely to include everybody. For the first study, we will have subsequent expansion of the protocol, for example, hopefully look at additional studies depending on how the first study goes. So it's, I know it's not satisfying, but it's difficult to me to say with certainty what disease stage would be included in that first trial. But as soon as we have that information and the protocol finalized, as I mentioned, it will be available on clinicaltrials.gov. And then we would also include the communication to the patient community.
Got it. Jordan, there's a couple questions in here about, maybe what can the community do to help expedite this process, or their role in helping with this, prepare for the study. Could you maybe address that a little bit?
Yeah, I mean, I think that one of the most important things that people in the community can do is continue to, finish up any visits they have with the natural history study. Completing that study and having as complete of a data set as possible is really important. Some of the other things that the patient community can do is really connecting with each other and staying in touch with the advocacy groups, finding ways to raise awareness of the disease in order to, really make sure that we're thinking about the entire patient population and that we understand the prevalence of the disease as much as possible. So if there's any patient registries that are developed, then participating in those is very important because this is such a rare disease. Every patient matters, right? And so every new diagnosis matters. And we want to make sure that, the patient community is aware of those and is bringing everybody together. So I think that those are really good ways to begin to prepare, for a clinical trial. And it will generally speaking, it will improve the options for the patient community as a whole, because, you'll all be working together and be connected. Does that help answer the question, Kristina?
Yeah, I think that's very helpful. Thank you, Jordan. There is a question about, roughly how many patients will be in the clinical study. And I know Kemi spoke to the fact that, the first study will be a safety study and it will be a small study, but I didn't know if there's anything else you wanted to say about that, Kemi?
Yeah, so the first study will be small. And I can tell you that it's going to be fewer than 100 patients, but we don't have a final sample size in terms of the number of patients in that first study.
Sorry, I'm scrolling through. I think maybe, Jordan, could you speak to the cost of participating in a clinical study for families?
Sure. So in Ionis's clinical trials, there is no cost to participating. So there's no cost for drug, all study related assessments and healthcare costs are covered by the sponsor of the trial. And any expenses that are incurred due to travel, such as parking or airfare or hotel, are typically will typically be reimbursed. So expense shouldn't really be something, I think, that individual participants or interested participants should be worried about. I think that some of the things to think about when you consider participating in a clinical trial are kind of those secondary costs, right? So the costs of, thinking about, the loss of, work hours, right? I think that that's something that people sometimes underestimate, right? How it can be, the amount of time spent, traveling to clinic. Those are things to think about before you agree to participate in a clinical trial. Childcare for other children is another thing to think about. So, participating in a clinical research study is really is a huge commitment. And the people that do volunteer to participate, they're very much the heroes in agreeing to participate and agreeing to put their own families and their own lives kind of to the side so that they can participate in this experimental study. So we recognize that it's a commitment. Staying in the study is, something that's completely voluntary, but is also something that is really important to make sure that we have the data that we need. So, it really is a commitment, and people should think really carefully about it because it goes beyond, what the costs are to directly participate. It goes to, what the cost is for your family and your community. So I think, kind of elaborated there, but I just wanted people to have a good understanding of, what it looks like to participate.
Absolutely. And it's, go ahead.
Is it, I'm sorry, because I'm getting some messages. So thank you guys so much for the update. I think a lot of the big questions we're unable to answer right now. When do you guys anticipate having more information, and how will that information be communicated to families? Because a lot of families feel right now they don't have enough information to plan for the future. So they don't know, there's just a lot of unknowns, especially for like mid-stage and late stage patients. And so do you know when you would have more information, and just so they can have some guidance and like plan out, how they should live the next couple of years or what they should anticipate?
I can go ahead and try to, if Kemi, or I can go ahead and try to answer that. I think that it's really difficult right now for us to anticipate exactly, a month or an exact date when we would anticipate a clinical trial starting. Like Kemi said, we do anticipate that it would start, at the earliest, second half of 2022. So that can be, right now we just don't have additional specific timing. But we recognize how difficult it can be for families to try to live in that limbo. And so when we do have additional information about timing that we feel comfortable sharing, because, we don't feel as though it will change, then, we'll be in a place where we can share that. Kemi and Kristina, do you want to elaborate?
Yeah, and I think, our hope is to make sure that we're sharing realistic expectations, right? We don't want to give you put a stake in the ground on a time that we may not be able to commit to, and it may be for reasons beyond our control. I think maybe, Kemi, there's a question about expediting the development process due to the severity and progression of the disease. Maybe you could speak to that a little bit, and that'll give a little more color on what we are making sure we're considering.
Yeah, so there are expedited pathways that you can use once you have data available, from the study, but you really need that data in the clinical data in patients to be able to make a case and qualify for that. Because Lafora disease is an orphan disease, so a rare disease, it does allow us to get timely scientific advice from the FDA and EMA, for example, to help us to plan the study in a way to maximize the chances of success. So Ionis has used, every opportunity, and we'll use any pathways that we can to expedite getting this potential therapy to patients. But as you mentioned, a number of times, unfortunately, as far as initiating the first clinical trial, we can't commit to a date earlier than what we've communicated right now. Just knowing all the steps that need to be done to initiate that first study.
Absolutely.
Sorry, Kristina, one more about placebos, because Jordan has done a good job in the past answering this question for us, but why is a placebo necessary, and what could the advantages be of that for rare diseases?
So I wouldn't say that a placebo is necessary. I would say that it's preferred by regulators. So we could certainly, plan a study that FDA doesn't agree to and take a risk, but it's also a huge risk. Placebo is preferred because it's a clean comparison and it's easy to design a statistical analysis to look at the difference between a treatment effect versus a placebo. So for example, when you use natural history, no matter how good your natural history data is, there are going to be certain elements that are confounders. So they can bias, the interpretation of the data, and that's what regulators like the FDA are concerned about. That's why they prefer a placebo comparator. That, of course, is, the preference of any company that's looking for therapies for devastating diseases is not to use a placebo. Obviously, we want to, give a potential therapy to every patient and not, give a placebo. But, we're limited by regulatory processes. And, I think that one thing that we can do, and we definitely are planning, is to enable every person that participates in the clinical trial to have the opportunity to get an active drug at some point during the trial, right? So to design it in such a way that if placebo is used, the period of being on placebo is minimized, and that you have the opportunity to get an active drug during the study. But, beyond that, I just don't want to give the misconception that placebo is off the table, because it's not, unfortunately.
Yeah, and maybe we could just, I think, share just some insights in our experience in other rare, ultra rare diseases. While it's very difficult, it's difficult to watch, right, and have that placebo arm, but there have been instances where I think SMA is a great example. The phase 3 study that we had to run in those patients. Actually, because there was a placebo, you saw that clear differentiation between treated patients and untreated patients, and that actually allowed the study to stop at an earlier time point than was anticipated. So, there's benefits to having that clear differentiation between a placebo control and trying to compare it to the natural history, which could be much noisier and harder to distinguish between the two groups. Sorry, looking through more questions here. There was also a question, Kemi, just about how this treatment may work by targeting the GICE1 and the Lafora bodies. Do you anticipate this being potentially playing a role in stopping disease progression? And do you think that there may be an effect on the existing Lafora bodies based on the preclinical data that we've collected to date?
So I can answer that question based on the preclinical data that we have, right? Because we don't have any data in humans. And we do have evidence that giving this particular drug can stop the accumulation of Lafora bodies. So Lafora bodies, as I mentioned, accumulate throughout the nervous system, including the muscle, the nerves, and giving the drug can stop that accumulation and progression. We have to see whether that's the same in patients, and more importantly, whether doing that halts the disease and possibly reverses the disease as well. That's why you perform a clinical trial, because you hope that the findings in animal studies would translate to humans, but you have to do the study to find out.
Thank you, Kemi. There's a couple questions about how do you indicate your desire to participate in those decisions. And I think it's important for us to, that's something that you could communicate with your physician, that you would be interested in that, as well as once there are study sites available and once there's a study designed and it's posted on clinicaltrials.gov, that would be when you would be able to reach out to the study investigators or the study physicians and let them know about your interest. And ultimately, Ionis will not be deciding who is in an actual study. The investigators that are running the study will actually make those decisions themselves. And I think I've tried to go through to see most of the questions kind of overlap with many of the things that we've spoken to already. I think, on getting more information, it's really important to work closely with the patient advocacy organization and Chelsea's Hope. And we're committed to making sure that we provide up-to-date information to the community. And, additionally, you can always reach out to Ionis at the patients@Ionis email address as well. And I think I'll toss it back to Jordan to see if there was anything else that we may have missed, because I think we're coming to the hour. And I just want to give us a second to make sure we covered everything.
Yeah, one last thing that I think we didn't get a chance to cover was there is a question about the delay in the start of the study. So I know it was previously communicated that the study was anticipated to start in this year, in 2021. And in June we released a community statement where we communicated that the study was going to now be starting at some point in 2022. So I just wanted to give Kemi and Kristina, and we can all kind of contribute to this. What are the kind of the reasons behind that? What can we share about about why there was a difference in communication between last year and this year?
So Jordan, what I would say about that is that delays in drug development, delays are inherent to drug development. And, many times, we plan for certain series of preclinical studies and events and interactions with regulators to be able to start a study, but delays just unfortunately happen. We're currently in the middle of a pandemic, which has affected many clinical trials. So it's hard to give you one particular reason, but we do know that the date that we're communicating for an anticipated start is realistic right now, and we don't want to give false hope and start retracting statements down the line. So it's hard to give you one particular reason, Jordan, but I will say that we're trying our best right now to make up for time.
Thank you. So I just want to thank everybody, Kemi and Kristina and Mathias and Gail, everybody on Ionis for participating and sharing so much important information, and Chelsea's Hope as well for helping put this together. So Lena, do you want to close this out?
Yeah, well, just to reiterate what you're saying, thank you for the whole Ionis team. And Kristina and Jordan, if anybody has any unanswered questions, they can email. Could you go back to the last slide?
Oh, of course.
Thank you. They could definitely email Chelsea's Hope. We're in communication with the Ionis team, or you could also reach out to Ionis directly. They've been really great at communicating directly with patients. So thank you guys so much for your time. Thank you for this update. We wish there was more, but we're hopeful, and we can't wait for when that information will be released. So thank you so much for your time, and thank you for the participants and everyone submitting their questions. Yes, thank you, and welcome, Kemi and Mathias. We're excited to have you guys on the Lafora team at Ionis.
Thank you, Lena.
Thank you.
All right,
Thank you.
everybody.
Thank you. Thank you for working with us on this.
Bye-bye.
Bye.
Bye-bye.
Bye bye.